Okay, in this video tutorial we're going to look at fungi. So fungi, you've all seen fungi before. For instance, in this picture, often the furries that grow on food when it's gone bad are basically fungi. But there are some different types, and some of them are true microorganisms. That means they're actually microscopic, and some of them are much bigger than that, so technically speaking don't quite fit into that, but we often include them anyway. So there are different types of fungi. You'll have all seen them before. Three main types: molds, mushrooms, and yeasts. So molds are the furries, so when food's got that kind of furry stuff growing on it, that's molds. Mushrooms are essentially just a much larger version. They're just much larger. Similar structure actually to molds, just much bigger. And yeasts are the single-celled ones, so these are the true microscopic ones. Single-celled, used in baking bread and things, so you've probably heard of yeast as well. So, fungi are all heterotrophic. That means they can't make their own food, and that's really important because fungi were historically actually considered to be plants or grouped with plants. But there's a key difference there, that fungi don't make their own food. They don't photosynthesize. And so they're actually in their own category. In an evolutionary sense, plants evolved from cells that had the ability to photosynthesize. We think they evolved originally from bacteria that had pigments that could absorb light energy and essentially carry out a basic form of photosynthesis. However, fungi are heterotrophs. They're essentially consumers. They eat material from living or dead organisms. And fungi actually play a very important role in terms of that, in terms of feeding off dead organisms. So without fungi, fungi are often considered a type of decomposer, especially molds are one of the key decomposers. And without those decomposers, if dead material will essentially build up all around us, there'll be one consequence. But the other thing is that if there weren't decomposers like molds, nutrients would be locked or trapped in those dead things, and so there wouldn't be enough nutrients being released or cycling back to plants. So once something dies, decomposers break it down, and some of those key nutrients that are locked up inside it are released, and plants can use those to grow, and so they keep cycling around and around. So we often refer to that as the nutrient cycle. So decomposers like fungi, like molds, are really important in that nutrient cycle. But key point of difference there, they're not plants. They can't make their own food, or they're heterotrophic. They feed by extracellular digestion. So we looked at that in the video on bacteria. Essentially, they secrete enzymes, and those enzymes break down the food, and then they absorb the nutrients or the chemical components of that food once it's been broken down. They can be unicellular, means one cell, or multicellular, made of many cells. So yeasts are our unicellular, and there are different types of yeast. So we often refer to baker's yeast as a species that's used for baking, but there are slightly different types of yeast, but they're all single-celled. Or multicellular, things like molds and mushrooms are made out of lots of cells, and they tend to be the macroscopic, the ones we can see without a microscope. So let's start with molds. Most fungi we can see are molds, multicellular. So things like the furries on the strawberry, or here's a coffee cup. I don't actually drink coffee, but a colleague left this on my desk. And you can see after a while it had a lovely collection of molds growing in there. So some beautiful colourful molds. And they do, our instinct is to look at them and go, ugh, yuck, because that's evolved so that we don't eat food that's bad. But molds, like I said, play a really important part in that nutrient cycling, and some of them also produce really important things for us, like one type of mold produces penicillin, which was the first antibiotic, something that kills bacteria. that was essentially discovered. So molds aren't all bad either. If we look at the structure of molds, the reason they look so furry is there's lots of these thin thread-like structures, and on the top of each of these thread-like structures is kind of a ball called a sporangium, and that basically contains the spores. So these spores can go flying off and if they land on an appropriate food source. So again, just like with bacteria, molds need a certain amount of moisture, a certain amount of nutrients, the right kind of pH. So if one of those spores lands somewhere where it has all those things, it will then grow, it will germinate essentially and grow into more mold. What's different between bacteria and fungi is that these spores, they often can survive in very dry conditions. So the mold can't grow without water, but it can survive in kind of a dormant in that spore form. Some bacteria actually do the same thing. They make a special little spore, they make a little capsule around themselves, and that way they can survive dry conditions. But this is one reason why a lot of dry foods, so things like cereals, flours, all those sorts of things, a lot of dry foods that you'd have in your pantry will have a few mold spores in them, and that's not a big deal, but it means if they suddenly have a little bit more moisture available to them, if they get a little bit damp, usually the first thing to grow on those dry foods are these molds. And so that's one reason, for instance, bread tends to go moldy rather than having bacteria on it. So here's the structure of a mold. We've got sporangium, which release our spores. So you can see an open one here. Sporangium releasing spores. The stalk-like bit here are often referred to as hyphae. A collection of these threads is sometimes referred to as mycelium. And then we have feeding hyphae, are almost like, look like roots of a plant, but very different function. Remember, roots of a plant only absorb water, maybe a few like ions and minerals. But here, these actually feed. They actually release enzymes that break down the thing that they're growing to. So they look like it, but very different function. So those are the key parts of a mold. So there you go. Sporangium is the head that produces spores. Hyphae, the thin thread-like structures that make up the body of the mold. Feeding hyphae, the ones that penetrate or grow into the food and carry out the extracellular digestion. And a spreading mass of hyphae, so a whole bunch of them is often referred to as mycelium. In terms of mold growth, it's pretty simple. They grow asexually. So what happens is they release spores. The spores land on a suitable food source. They grow into the full mold structure, and then they release spores again. So these spores aren't like eggs. They have the full set of DNA. So essentially, it's just a little cell that buds off and can grow into a full-grown mold. And sometimes the molds can actually sort of grow by vegetative growth, I believe. So as the mold grows, it's usually all connected. But if it breaks, then essentially you'll get two molds. They can split. So yep, once they've grown, they release spores, and you can see there's kind of a cycle. So the fully grown molds release spores. Spores land on food, grow into produce the mycelium. And sporangium release spores, and we keep going round and round. So what do molds have to do with the Salem witch trials? I just thought this was a really interesting thing that I learnt about when I was at university. So the Salem witch trials, if you didn't know, Salem is an area in America, and in 1692 there were a lot of witch trials. So and that was due to a lot of reasons. People were quite superstitious at that time, but at this time a lot of people were accusing other people of being a witch. And unfortunately, the standard way to test whether someone was a witch was to tie them to a chair or lock them in a cage and put weights on it and essentially dunk them into a river. and if they drowned, then they were not a witch, and their honour was restored. And if they did not drown, then they were a witch, in which case they would be burnt at the stake. So if you were accused of being a witch, and there were enough people who were accusing you, the outcome wasn't very good either way. I guess if you couldn't hold your breath, you'd drown, and if you were pretty good at holding your breath, then you'd probably be dunked a few times and then burnt at the stake. But scientists have sort of wondered why this was sort of going on at the time, and there is some evidence that suggests there was a mold growing on wheat at the time that produced a toxin that was similar to ecstasy or some other hallucinogenic drug. So the theory is that one reason why there were so many of the trials in Salem, in this part of America, why they were particularly affected, is possibly due to the fact that people were eating the bread, and the bread contained this hallucinogen, made them hallucinate and see things and act a bit crazy, because essentially they were drugged. And then so if people saw someone acting a bit funny and talking crazy and running around naked or things like that, because they were drugged, they would assume that maybe that they were possessed or a witch or something like that, and suddenly they'd be accused and burnt at the stake or drowned in the river. So there is evidence that suggests there was a mold growing on the wheat that may have contributed to it, which I find really interesting. One reason why I think there's much stricter controls and things on wheats and things these days. So, secondly, mushrooms. Mushrooms are basically just fungi that are big enough that we can see them, and big enough even to eat. And they have a very similar structure essentially to molds. So the stalk here, like that hyphae, feeding hyphae into the ground, and the bit on the top is essentially like the sporangium. The structure's a little bit more complicated than that. Often they have like these sort of gill-like structures on the underside, and the spores are released from within there. So usually on the underside of the mushroom that you eat, there's that darker patch. Well, the darker colour is actually the spores on the surface of those sort of gills, just on the underside of the mushroom. There are some mushrooms that even glow in the dark, and then there are some mushrooms that you can only eat once, because they contain toxins, and if you eat them once, that's kind of the end. Yeasts, our third group. So these are basically unicellular. Now, one thing my students often get confused about is they think of yeast that they use for baking. Now, each of these little granules is not a single yeast. Each of those granules is basically a little cluster of probably thousands and thousands of yeast cells. So each little granule is not a yeast cell. It actually contains thousands of tiny little yeast cells. So yeast are single-celled organisms, living things, and you can see here the yeasts have these little buds, these little bits sticking out, and that's how they reproduce, very much like bacteria, except one difference: rather than growing in size and splitting in two, they get a bud forming, so a smaller bud that forms, and this grows in size and then eventually sort of buds off, and that's what we call the daughter cell. And so it's a little bit different than the bacteria. There's evidence now that there is actually often slight differences between the daughter and the parent. For instance, cellular junk that accumulates in the cell as it ages is kept in the parent cell. So the daughter cell often lives longer than the parent cell from which it was formed. But genetically they're identical, so they're identical. So it is a form of asexual reproduction, and it is just what we call budding. So yeast divide like bacteria. It's a type of, I guess, binary fission, or often referred to simply as budding. So the one key difference here, rather than simply growing in size and splitting in two, the daughter cell buds off. So we have a cell that buds off, and it usually starts off slightly smaller than the original parent cell, and genetically identical, but there are a few differences, just like the size, or like I said, the junk that's accumulated in the cell over its lifetime tends to stay in the parent, and so the daughter cell lives a little bit longer. So that's it for basically for fungi. So the key thing to remember: the three types: mushrooms, molds, and yeasts. Yeasts being single-celled. They're all heterotrophic. They can't make their own food. They feed off other things by extracellular digestion. Yeasts grow by essentially binary fission, or budding rather. And we've looked at some of the key parts of the mold there. So the sporangium, the spores, feeding hyphae, parts that you might be asked to identify in a test. Cool. I hope you found that helpful. And we'll get on to the next one.